Chemical digestion in the alimentary canal occurs by the presence of specific enzymes that are secreted into the lumen of the canal. Undigested starches, proteins, and lipids are molecularly too large to be absorbed through the cells that line the surface of the mucosa and into the deeper capillaries that are found in the lamina propria and the submucosa. So enzymes must break down those molecules into their component molecular parts before those can be absorbed into the bloodstream. Enzymes are proteins that function as catalysts, where a catalyst contributes to a specific chemical reaction. Each enzyme catalyzes a very specific reaction within the body, and the body has thousands of different enzymes. Enzymes must first bind with their reactants, which are called substrates. The enzyme will align the substrate precisely in a way that allows the chemical bonds to more easily be formed or more easily be broken between the substrates. Digestive enzymes function to break chemical bonds between large complex molecules, which separates them into simpler components. After the reaction has occurred, the enzyme releases the changed reactants, which are now called products, and the enzyme is available to catalyze a subsequent reaction with fresh substrate. Figure 2.27 examines the basic way in which enzymes work. This particular enzyme is binding two substrates, aligning them, and a chemical bond can now more easily be formed to link the substrates together. All the enzyme did was to align the substrates in a way that they can chemically react with one another. At the end of the reaction, the enzyme is unchanged and releases the product. The enzyme is now able to accept a fresh set of substrates and complete the reaction again. Digestive enzymes perform the opposite that is seen in this diagram. They take large, complex substrates and break them down into smaller and simpler components. Here we will summarize the major sites of chemical digestion by enzymes within the alimentary canal. The mouth begins carbohydrate digestion with salivary amylase and lipid digestion by lingual lipase. Both of these enzymes are produced by the salivary glands. The stomach acids will destroy the salivary enzymes when a bolus of food and saliva reaches the stomach. So carbohydrate and lipid digestion will temporarily stop within the stomach, but the stomach does begin protein digestion as it secretes an enzyme called pepsin. The small intestine resumes carbohydrate and lipid digestion in addition to continuing protein digestion. The small intestine is the final site of chemical digestion for the carbohydrates, the proteins, and the lipids. Carbohydrates are digested to the monosaccharides of glucose, fructose, and galactose by pancreatic amylase from the pancreas, in addition to other carbohydrate digesting enzymes that are already present in the small intestine. It digests lipids to fatty acids by the presence of pancreatic lipase from the pancreas, and it also digests proteins to amino acids by trypsin from the pancreas, in addition to other protein digesting enzymes that are already present within the small intestine.